Now we will enter into very important topic in our drug design process is the virtual screening in drug discovery. It is based on the chemi-informatics approach. Of course, we are going to use the bioinformatics also. Without that, we cannot proceed. You see in this particular slide, you are finding some of the chemical structures on the left side of the screen. In the right side of the screen, you have got some of the biological materials, that is proteins, which are present. So these two are, we are going to start. The first one is going to get from the chemi-informatics tools. The second in the right side in the protein, we are going to get it from the bioinformatics tools. And whatever the tools which are present in the database at present now, if you see them, it is having more than 10 to the power of 20 chemicals are available to be tested for the drug synthesis. Similarly, if you go to protein level, we are going to have a 10 to the power of 4 to 10 to the power of 5. It means around 1 lakh spaces, biological spaces, proteins are already available in the database. So we have to screen all of them in a combination when you are going to have. When these each one is going to react with all chemicals which are reacting, it is going to be highly cumbersome in the HTS screening, what we have already observed yesterday, combinatorial chemistry. That is the reason we are going to do virtually, that we call it as a computation to screening, where we use the computer for the experimental screening. We want to avoid it. So what we are going to have is, we want to test all the available compounds in the validated targets. What is the validated target? The target which is identified to have a interaction site which is already been known. The chemical structure of the protein is already known. So the compound which is having a features that is also be known. So we want to study them in the virtual screening process to pick up the moiety which is having a worth considering for the drug design. So the first journey in this particular case, first step is choosing the right molecule. So then only we are going to have a success. So the goal is to choose the lead compound that can be optimized to give a drug candidate. For that purpose, we are going to do a optimization. So the optimization by using a chemical synthesis to modify the lead molecule in order to improve the chances of being successful drug. We modify all the functional groups and their sizes and electron intensities and polar surface area and considerations of Lipinski rule of 5 rules. Then we optimization, it will lead to a good moiety. So, but the challenge is the chemical space is very vast, where we have got 10 to the power of 20 chemicals. And the estimates are going to come very low, which are number which is less than the 10. What we want a molecule, it can be 2-3 molecules we want. The best one with the highest scores. So it means that 1 billion compounds, we need to have a 13 heavy atoms which fall the Lipinski rule of 5. And there are 65 million known compounds which are available in the chemical database PubChem. A typical pharmaceutical compound collection contains 1 to 5 millions. Remaining are, we call them as the reagents or the reactants or starting materials, and the toxic chemicals, which we don't consider them for the drug synthesis. So 1 to 5 million we are going to consider. Similarly, high throughput screening allow up to 1 million we can do. The number of compounds can be tested. But very small portion of the available compounds are there. Large scale screening is very expensive if you go like this. So in thousands which are present, then I put throughput screening is good. But when you are going to the lakhs, it is going to be expensive. So reason is not all targets suitable for the high throughput screening. Reason is we don't have detection methods. We cannot determine the complex formation with each. That is not feasibility there. So externally, the stability issues may be present, and the sensitivity and interferences in the analytical assays, whether it is cell-based or biochemical. So these are all the things which are making a complex in the high throughput screening. So for that purpose, we are going to use a virtual screening. So we take the screening for either you want to have a HTS or by the ritual. We are going to start first with HTS only because we want to have a known compounds which are present from the combinatorial library, and we are going to screen. That is one journey. Or we can go to the virtual screening and take a all the moieties which possible manner, and we are going to screen them. So these are all inputs are going to come from the databases. So this one is going to come from a protein, target protein, where the 3D structure is already identified. So when you screen them, we are going to get a lead, and lead optimization is going to result in a drug. This is our end journey. So the basic goal of the virtual screening is the reduction of the enormous virtual chemical space to manageable number of the compounds that would inhibit the target protein responsible for the disease, and also have the highest chance to lead the drug candidate. That is the reason we are virtually doing it. Otherwise, we do the HTS only. So the virtual screening, we are going to have two stages. One is unknown and the known, and the same story will begin, and the similar answer we are going to write here also. Either if it is 3D structure is unknown, it is ligand based, and the 3D structure is known and structure based. In the structure based, we are going to have a docking studies and finalize a molecule with the good scores. In the ligand based one, we are going to do two major approach. One is a similarity search, where we are going to have a COMFA-COMSIA analysis, where we are going to have a similarity, and we are going to have a pharmacophore mapping by using the SAR and QSAR. And ultimately, what we are going to do to find this active from the inactives, which are known, we need to use a machine learning tools so that our journey is simple and the fast. So we are going to use a artificial intelligence. It means that depending upon the structural and biological data available, one or more active molecule known perform the similarity searching. Several active known try to identify the common 3D pharmacophore so that they can result in a similar response, because they are occupying the similar 3D space. And a reasonable number of active and inactives we can train in the machine learning model. Inactive molecule, when we are tried in the machine learning, it will give what are the not drugs. Active molecule, what we are training, that will give identify as a drugs. So when we are training it, we need to take both failures also, success also, what we are having in the machine learning tools. So that our new moiety, what we are giving in the virtual screening, that will easily identify, because it is already got trained, whether it is going to have a activity or not. So the 3D structure of the protein is known, and we can use the ligand protein docking, and finally we confirm whatever it is suggesting by the machine learning. So the machine learning is going to work. That is the reason it will get a success in the docking also. There is a hybrid mode we can have. So in the hybrid mode, what we are going to have, we will start with a database, save the similarity. So, and prepared databases, and we optimized structures we take for the pharmacophore. Or we are going to take the structure-based pharmacophore from the docking, and we are going to do the screening with respect to that particular pocket. And ultimately, what we are going to use in the docking studies, where we are going to have a gold glide ICM, and the post process we are going to use a C-score and solvation corrections, and potential lead molecule we are going to do. So what are the solvation corrections and potential corrections we need to do? So for that purpose, we have got two approaches. One is the cleaning of molecule, another is the filtering of molecule. So what is the cleaning of molecule? In the cleaning of molecule, we need to see that our toxicity cannot cross certain moieties which are present in the chemical that will cause the toxicity of our body. We know that halogen, certain types of reactive chemicals, heavy metals, they cause the poisoning and cumulative poisoning in our body. So, and radioactive chemicals, they are very toxic because they are having a cell death. That is the reason we remove the isotopes, salts, and mixtures, and we take the protonations and make the normalization so that all the assumptions are going to be correct, and remove the duplicates and invalid structure so that our time will be reduced. This is what we call it as a cleaning of molecule. This approach our visual screening process with less time and without having a duplication. There is another approach which we use in the visual screening is we have to use the filtering of a molecule. So we are going to have a huge number of million of molecule to come down to less number, 5 million to 1 million, 1 million to 10 lakh, that is 5 lakhs like that. So the filtering molecule, we are going to use a defined filter where we don't find certain aspects and factors considered not to be working in that particular receptor. So we don't consider them. So we filter. So another one is we remove the problematic moieties and the pains. That's what we are going to do now in subsequent slides. So we consider only frequent hitters, that is having a good ability. So then physicochemical properties descriptor calculation, and we will get the similarities. Based on the similarity only we will pick up, and remaining them we will filter it. Ultimately at the end we are going to opt the protonation at 7.4 pH because our drug molecule is going to work in our body at pH 7.4. So in those cases, then we are going to simulate the pH of our moiety to 7.4, where your pKa values and dissociation constants, what you are going to have, that will fall into the Lipinski rule of 5 or not, we will be having. Based on that, we are having a solubility also. So ADME will also be considered. So ultimately, this approach is going to solve our absorption problem also. Ultimately, it is resulting in a drug. This journey is making a foolproof to us. Now, ultimately at the end, we are going to attain the properties the drug minimally it should have so that we can take it further for the clinical trials. So what are the properties which we need to consider when you are considering the drugs filtering, after filtering and the cleaning of molecules? We have to see the drug-like properties all those molecules are having or not. Then only we take them. Otherwise, we will drop there only. So what are the properties we consider we have to see now? So the first one is the structural properties. That is hydrogen bonding, where we have got proton donors, where we are going to have not more than 5, and proton acceptors not more than 10. Polar surface area not less than 140 angstrom square. Lipophilicity, where log p value 5 less than 5. The shape, where we are going to have the depending upon the what the pocket is present. Molecular weight, obviously less than 500. Reactivity and pKa, that is also very much important. So we need to consider, because most of the drugs are weakly acidic and weakly basic. At 7.4 pH, how they behave, we need to consider at the end. The second one we need to consider is physicochemical properties. In the physicochemical properties, first thing we need to consider is its solubility, water solubility and lipid solubility. And once the solubility is completed, then we need to see the permeability. So because it is need to have a both soluble also, permeable also, so that it will be having acting as a functional as a drug. And you may have in the doubt, if it is falling in the BCS class 2, what is required to be, can we drop the moiety there or what? So most of the times in the BCS class, which category they are falling, we need to identify at this stage. The permeability and solubility issues can be resolved at the R&D stage itself or at the formulation development stage by the enhancement of the solubility and the formulation approaches for the increasing the permeability. At the end, what we need to consider after these two is whether the moiety is going to have a chemically stable or not. Even though you are having a solubility permeability criteria as met, if they are not stable in the given environment, so it is worthless to consider further. So this property also we need to see. Next, after the structural properties, physicochemical properties, we need to see the filtered and the cleaned molecule. Does they carry biochemical properties? So whether they undergo the metabolism phase 1 and phase 2, cytochrome P enzymes, they are involved in the metabolism, whether they are going to get degraded. If they are not getting degraded, they are accumulated in the body. Then they act as a poison. So another one, what we need to do is we need to see that whether it is protein bound or tissue bound. So this is going to tell you our half-life and residence time in our body. Next one is what we are going to do the transport system, whether it is going to have a passive transport or active transport, it is depending. Most of the times the passive transport you can have with a concentration gradient, you can have it, but with active transport, it will have a nonlinear kinetics, Michaelis-Menten kinetics. That is the reason you are going to have a saturation mechanism. Till the saturation, they follow the linearity. After that, they go to plateau stage. So the combined approach, whether they are present, we need to see as a biochemical property before considering it as a drug. Ultimately, at the end, we are going to see the pharmacokinetic properties and toxicity. So what are the pharmacokinetic properties? This is clearance, half-life, that decides your dosage regimen. So number of times you are going to give. Generally, we repeat the dosage at every half-life. That is the reason. If the half-life is high, then you are going to have a number of doses per day going to reduce. So another one is a fraction of the dose absorbed, that is bioavailability. It is appearing in the rate and extent available in the systemic circulation. That also we are going to see. And drug-drug interactions also very much important, because most of the diseases, they are co-occurring, like pains and fever, we are going to have simultaneously, and both the drugs combinedly you are supposed to take. So such minimal things we need to see whether they are going to have BP and diabetic drugs. Preferably they will have a drug-drug interaction. If they are having both, is going to get affected there. So ultimately we are going to have a LD50 values. What is the dose at which 50% of the population dies? So if the dose is very low, it means you are having high toxicity. The LD50, the lethal dose 50 is very high, very, very high. It is having grams. So then you can have that, attaining that concentration in the body is negligible and you don't have any toxicity. This will give you the idea for you to whether to consider the drug as a drug-like moiety or not, and you can drop the knob itself, or you can modify in the R&D itself. Now, if you want to take dielectric drug-like moiety, you won't get, because you are going to coming from the 10 to the power of 20 chemical databases. You need to take lead-like. Don't get confused with lead-like and drug-like. So if you are saying drug-like, and you will drop all the chemicals, whatever it is available in the chemical database, they will not be drugs, but they can be leads. So we are missing some benefit by considering only drug-like properties. Because we need to take some of the chemicals which are acting as a lead, we can modify later, convert into a drug. That is the reason we need to take leads also. So till now what we have seen, the drug-like molecule, like molecular weight less than 500, log P values less than 5, hydrogen bond donors less than 5, some of the hydrogen bond acceptors, that is less than 10, polar surface area 140 angstrom square, number of rotatable bonds 10. So the last two are the Weber's rule of Weber's rule. The first 4 are considered with Lipinski rule of 5. So these are all important to exclude the reactive compounds. They are the frequent hitters and warheads if you consider this criteria. Whereas the lead-like molecule will not meet this. That we need to consider very much during in early stages. So what the lead-like molecule will have a characteristic now we need to see. So the first characteristic in the lead is molecular weight 200 to 350. Sometimes it may go at 100 to 200 also. Later on, during the optimization, it will go to 500. And log P values 1 to 3. So less than 1 to 3 is okay. And single charge present, that is secondary or tertiary amines are generally preferred. And importantly, exclude the chemical reactive functional groups. So they are going to cause the toxicity. We need to exclude reactive compounds. So that is having a free radical like structures, so such things which because they are the chain initiators also, and they cause the carcinogenicity also. So such reactive chemicals we need to drop now itself in the leads also. Non-substrate peptides are also suitable. We need to consider because now the impetus is going on with the small peptide molecule to act as a drug in place of a biological molecule that is bigger, and your chemical molecule that is completely chemical compound. So in between we are going to have little bit higher molecular weight, not as that of the protein. We can consider the peptides also for the receptor binding during the leads. We can consider. Now, what are the things which we need to consider during your visual screening for the filtering of the molecule for the structural properties? So when you are filtering, we are going to have the basic washing of molecules we are going to do with respect to the following parameters. Like first you need to remove the salts because the salt form is ultimately going to come before formulation. Because what type of the salt, whether it is hydrochloride or sulfate, mesylate, mesylate or proxetyl, acetyl. So these are all things will come during your journey when you are making a formulation development where you have got a issue of solubility and permeability. So remove the salt at this particular juncture. And unwanted elements, when they are present, they are not easily available and they are not difficult to get in nearby vicinity. Or they are very difficult to rare earth chemicals which are present. So they are very costly and not available in all countries also. Such things better to drop now, unless otherwise you don't have any option. Filtering out the cationic groups also are very important because they are going to have changes in their polar surface area based on their presence. So calcium, sodium, potassium, such compounds you have to drop down. Filter out the metals like titanium, chromium. So most of the times you have got zirconium, neodymium, molybdenum. So there are rare palladium. They are similarly you are having a gold and silver. So these are all very making costly your compound, even though they get success also. Making them it is very non-affordable. So better to drop now itself. The rare chemicals, costly chemicals not available in the vicinity, such metals we have to drop it down. Often the salt filter keep the largest molecule in the data file entry. Yellow the elements like hydrogen, carbon, nitrogen, oxygen, fluorine, phosphorus, sulfur, chlorine, bromine, iodine. They are all very good suits to us, easily available, easy to synthesize, makes our cost effective molecule. So these chemicals better to be incorporated and filter will be applied based on these elements. Check with the atom types by adding the hydrogen and check it on oxygen, nitrogen and C and their valencies are correct or not, because you are generating the structure with a C, it is having a carbon atom in the 4 carbon bonding capacity it has got. But you have got your structure which is having only 3 bonds are present, one side is left. That is the reason proper checking of the atoms valency and these are getting filled in that particular structure or not, you have to see. Otherwise, that existence of that moiety is not there. So because the structure itself wrong. Check the formal charge. That is also very much important. These filters, it will make you reduce your number to very small. And your time will also be less and your success rate is also very high. Now filtering of the reactives. Sir, what I can reactive chemicals, how will I know it? So the reactive chemicals, they act as a false positives. Most of the times these reactive chemicals interact with the proteins because proteins have got a problem of astringent effect. They will be getting precipitated with the harsh chemicals, corrosive harsh chemicals. So they mutate, they change their structure in the we cannot say in the primary structure, but secondary and tertiary structure, obviously they will get altered. So pockets will also get changed. So interaction scores are very high, but they are not feasible in the regular cases. So that is the reason we need to filter out the reactives. So the reactives like sulfonyl halides, acyl halides, alkyl halides, anhydrides, they are highly reactive. Halopyrimidines, halocarbonyl compounds. Most of the times you are having in these compounds the X and R. These two are the substituents. X will be generally used for the halogens like fluorine, chlorine, bromine, iodine, tosylate, mesylate, such thing. R can be generally a functional group like alkyl, aryl, heteroalkyl, heteroaryl. So they makes the molecule further considering when you are going to have a different combinations also. When these basic heads are there, they are considered to be highly reactive, so better to drop. Like a dicarbonyl groups, most parhaloketones, there you are having most of the times imines, epoxides, they are highly reactive, aziridines, thioesters. So these groups are sulfonates and phosphonates are also going to get a problem. Michael acceptors and heterosubstitute carbonyl compounds, most of the times they will be having a ketoenol tautomerism, and they will make their functionality based on the R group, whatever it is present there. Heteroatoms and hetero single bonds, when you are having a repeated atoms, when they are present adjacent to, you are going to have N, N, S, 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 O, O. So these are all the groups which are abridged between the main moieties, that will cause a little bit problematic for the electrophilic protein reactive false positive it will give. So you have to filter it out. Most of the times you need to filter the synthetic intermediate also, chelating agents also. So in this particular case, you have got a phosphates and phosphonates. So hydroxymates and thiol chelators, they are going to highly be reactive. Metalloproteinases, they are present. So most of the times, electrophilic suicidal inhibitors like serine, threonine, and cysteine proteases. So these moieties better to avoid now itself, so that they are not going to give a problem later, because they are non-lead likeliness and lead likeliness categories. We need to categorize now itself and filter it, weed them out. Now next, after filtering these reactives, and we need to see the paints. What is paints filter? Paints are pan-assay interference compounds. So the expansion form of it is a pan-assay interference compounds. They called as a paints. So we need to filter this pan-assay interference. When they are going to interfere with your assay moieties, they cause a misinterpretation of your drug. Better to drop now itself, otherwise your interpretation is going to be wrong. Another one is the problematic scaffolds. They have got their very high cost for their synthesis, many dollars they are going to have, like what you are seeing in this particular group. There are so many intermediates. Many pharma companies, they have dropped because the synthetic were not feasible and cost of making them is very high. Even though the drug likeliness scores are very high with them and not feasible, so we cannot make it. So we need to drop them. That is called as a problematic scaffold filters and pain filter. Now we are going to see the rule of thumbs, that is checklist. So the checklist, you are already having it. It is a repetition. Obviously, for this question, you are going to answer. And similarly, you are going to get a separate question in the examination also. So the rules of thumb for the hit selection and the lead optimization is, for oral bioavailability, you are going to have a rule of 5, that is Lipinski. And for the toxicity, you are going to see the log p values less than 3, polar surface area less than or equal to 75. So we are going to consider them. So for permeability, you need to see the polar surface area less than 120, molecular weight less than 500. Blood-brain barrier, we are going to see the polar surface area less than 70. So the solubility, specific solubility, we have to consider. So general developability, that is number of aromatic rings less than 3. it is better, less than or equal to 3, we need to consider. So this checklist, it will give the idea that whether you are going to have a drug developability, less toxicity, and ADME properties met, and you are going to have a success rates very high. So this entire slide, it will give you the idea with the comments which has been given with the rules of thumb what is present. So that we need to consider. After you are filtering it and removing the paints filter, everything, we need to do the similarity search. What is the similarity search? So chemical, pharmacological or biological properties of the two compounds will match. So either it is matching with chemically or biologically or pharmacologically when they are matching, then we can consider them as a similar for the treatment of the disease. If you see the two structures which are present in the screen, they are chemically similar to each other with functional groups with what you have got a amine that is having a R, that is only a deviation which is present with 2 methyl groups are replaced with 3 methyl groups. And the hydroxyl group is having a little bit diversion of extension with one CH2, so that you have got compensation with the trimethyl groups which are present in the structure. So in this particular scaffold, they reflect the similarity with respect to the chemistry. Similarly, when you are having a bottom, you are seeing two structures. They are pharmacophore, they are similar because they are having a sulfur also, amine also, 2 oxygen groups. Here on the left, you have got a 6-member ring. In the right, what you are seeing is a it is having a 7-member ring. So even though they are having that, but it is also having a pentyl group, that is cyclopentyl group, that is replacing there. It is having a topocologically similar. The two structures of the less similar chemically. What you are going to find it is seeing from the size of them is but different. Yet they have the pharmacological activity same, namely both are angiotensin converting enzyme inhibitors. They are already successfully be used, even though they are chemically different, topocologically different, but they are acting pharmacologically, they are same. So they are called as a pharmacophores. The first one what you are seeing is a leads and the hits. So now what is required for the molecular similarity structure? Till now what we have seen, we have filtered it, washed the chemicals with respect to your salts, and we have matched and we have seen the rules of thumb, and we have seen the similarity. Then we are going to have a similarity search. This part we will take tomorrow at 10:10 class. We will continue. So we will go with attendance at present. Sharing.